Awesome. All right. Well, it looks like we have quite a few people logged on, and so we will go ahead and get started. All right. Well, hey, everyone. Welcome to the first Family and Researcher Roundtable. This is our final segment as we move forward in this series. My name is Casey Craig. Many of you know me as Stella's mom, but for those of you that are new to our community, I am also the executive director for the Cute Syndrome Foundation. I have really been looking forward to today's discussion because this topic comes up over and over again in our family support Facebook page. Upon our child's diagnosis, we receive these genetic reports that might as well be written in ancient Greek. And it will list either de novo or inherited mutations. But most of us are really just wondering, what does this even mean? And how does this affect our chances of having other children with SCN8A? So today we will get to learn all about this from our legendary researcher, Dr. Miriam Meisler. Now, I know we're going to have lots of questions today, which is great. Dr. Meisler really wants us to ask questions and make sure we all come out of this feeling confident and educated. So we want to make sure that you guys have all your questions answered. So if you want to type all your questions in the chat box, I will keep an eye on the thread as we go through the presentation today. And then everyone's done a great job keeping cameras and staying off and staying muted. That way we can just have the best bandwidth and not have background noise, which I don't know. My background noise is my dog snoring right now. So if you hear that, that's Miss Claire Bear snoozing next to me. And before I introduce our amazing guest, I do want to thank our platinum sponsor, Neurocrine Biosciences, for sponsoring this series. Okay, so without further ado, we have Dr. Meisler with us here today. She is a professor of human genetics and neurology at the University of Michigan and has a long history of studying the human genetic disorders and mouse models that are relevant to human health, especially in the SCN8A space. All right, Miriam, would you like to tell us a little bit about yourself and then jump into the presentation?
Sure. Well, as you can see, I'm at the University of Michigan in Ann Arbor, where I've been for many years. And we got into SCN8A in 1995 was our first paper on SCN8A. Wow. And our introduction to SCN8A was through mouse models, actually, because I'm trained as a mouse geneticist. Mm-hmm. And I have to say that I don't do any clinical work, so I really can't address those questions.
Sure.
But we had a mouse, and the mouse was interesting, I thought. And so we spent almost 5 years trying to find out what was wrong with this mouse. And then the answer was SCN8A. And so ever since then, we've been working on it. So I'll just get started here.
Now, first, I do want to point this out. We were joking about this and talking about it before we went on. You guys have to see this. This is Miriam's license plate. So this is how much her heart is in our world. And that just cracks me up. And I think I might have to get one.
It helps me find the car in a crowded parking lot.
Yeah, I love it.
So I think I've met a lot of you probably as I've attended several of the symposia at the AES. So yes, probably know me. So let's see what we have here.
Okay. Is it? Oh, there we go.
Okay, so as you all know, as well as I do, SCN8A is a sodium channel that looks roughly like this if we make a cartoon version. And these are the mutations that were known as of 2021. I don't know why my screen is jumping around.
No worries.
So at the moment, there are almost 1,000 individuals identified who have mutations of SCN8A. And the ones in pink and red here are the severe ones. These are the gain of function mutations, so to speak. And they result in DEE, which I think is what most of us are dealing with. And the blue are more rare. They're loss of function mutations, and they result in a much less severe phenotype. So how has this developed so quickly? As I said, it was 1995 when we first found the gene in the mouse. And then it was not until 2012 that we found the first human mutation. And that was a very exciting. I got a call from Michael Hammer, who probably most of you know, and he said, we found the mutation, and so we started studying it. In fact, 2 years now, we made that very mutation that Michael Hammer had found in his daughter, and we made a mouse model, which was the beginning of our work on the disorder. Cute Foundation came in 2015, and we had a wonderful first meeting. And in 2020, we reported our ASO therapy for the SCN8A mouse. So we've gone very rapidly from gene discovery to human mutations to the possibility of genetic therapy, which is my field, as well as drug therapy, as you all know. Discovering the gene in 1995, finding the human mutations in 2012, and my lab has pretty much focused on what we could do about it now. We reported in 2020 that if we treated the mice the day they were born with this antisense oligo, before, that's not possible in human because you wouldn't know on the first day. But when we did that, we could protect them for their lifetime. So then our question was, would this work in an individual once the seizures begin? And this is something we have just worked out. And it does look as if there's no damage done that would prevent this from working. And I'll show you that picture of that in a minute.
Awesome.
And then the thing I'm really excited about is that we've been able to do a CRISPR knockout of only the mutant allele in this mouse model. And that is very effective. So we can't do that in patients yet, but I think the future is very encouraging in terms of genetic approaches. So here's our data on giving this ASO after we give it intracranially. Can't seem to go backwards. Yes. After the seizure, after the first seizure, in these mouse models of the Hammer mutation. And honestly, I'm not touching this, so I don't know why it's moving, but so the seizures begin at 2 or 3 months in this animal. Then we give the first treatment and we repeat it every month, as these ticks indicate. And you can see we're plotting here just survival. And the untreated mice, or the mice with the control, only live for 4 or 5 months. But as long as we keep this up, we're maintaining these mice through time. So certainly ASO treatments are one possibility for SCN8A mutations. But there is not, I must say, there is not a company involved yet. Well, there's some tweaking it, but it's not there. Oh dear.
And when you look at this line, so that's the number of survival. Are they still having seizures?
Oh, no, they're not.
Okay.
No seizures and no deaths.
Interesting.
So
Okay.
we're really encouraged. So the topic I understand that you're interested in talking about today is these mutations and the difference between a de novo mutation and a germline mosaic mutation. So I'm going to try to give you hopefully some new information. I know you're all very well informed and you may already know this, but please interrupt with questions because my presentation is rather short and the whole point is to answer your questions. So as you undoubtedly know, a mutation is a change in the DNA sequence. For example, here, one single base. Here it's, I've colored it blue in the normal sequence, and it is changed to A in the mutant sequence. And of course, if this happens in the coding region of SCN8A, it may be deleterious. Now, why do we get mutations? How do we get new mutations? Well, every time a cell divides, as in growth and adulthood, the DNA has to be copied so that each new cell has a full copy. And this is achieved by an enzyme called DNA polymerase that has to copy the whole genome, all those chromosomes, from one cell so that the second cell gets a copy. Now, DNA polymerase is an amazing enzyme. It's very stable. It's very good, but like everything in biology, it's imperfect. So DNA polymerase has an error rate, an intrinsic error rate. And that only, given the repair enzymes, which also function, you only see about 1 error per 100 million bases. That's quite impressive.
Mm-hmm.
However, we have 3 billion bases in the genome. So that means that every time the DNA is copied, there are 30 new mutations.
Mm-hmm.
Now,
I don't mean to interrupt here, but so would I be correct in thinking that those 30 errors could end up with like a gene for high blood pressure or something like that, but our kids just, if it was a de novo, our kids just happen to have the mutation on the SCN8A gene as one of their 30 errors?
exactly. Most of these errors don't make any difference because most of the genome is not genes. It's only 1.5% of the genome has the coding sequence for your proteins.
Mm-hmm.
So most of this is just noise. And if it just happens in one cell, perhaps that cell might die.
Mm-hmm.
So that's the end of the story. Most of these are neutral. And that's the source of all our SNPs. You may know about SNPs and all the variation in the human genome.
Mm-hmm.
But if they're in the wrong place, you know.
Mm-hmm.
So what if the mutation occurred in an egg or one sperm that happened to give rise to the new individual before fertilization? Then every cell in the resulting individual would have the mutation. And that's what we call de novo mutations. And this is also the case if you have a mutation that occurs very, very early in mutation. Then almost every cell will have it. And most individuals with SCN8A mutations are, in fact, de novo mutations of this type. And they're not mosaic. The whole individual. If the mutation is carried by the egg or the sperm, then the whole individual, every cell will have that mutation. And that's the usual situation.
How is there any way to know that, that for sure that it came from a mutated egg or sperm?
I'll get to that.
Okay, good. I'm getting ahead.
And then if not, please bring it up.
Okay, sounds good. Sounds good.
So these mutations, these errors due to the DNA polymerase can happen at any point in your lifetime. And does happen. We know now, we can see them, we can detect them. They do occur throughout life, whenever a cell divides. So, for example, if that mutation happened during embryonic development, not right at the beginning, but during development, then you would get a mosaic, what we call a mosaic individual. So you would have the sperm and the egg are perfectly good. The cells start dividing, and then the error occurs early. And then, as the cells keep dividing, the offspring of that cell will also have the mutation. And that will give part of the body that has a mutation. Most of it is wild type or normal, as we say. And that means that when you get the adult, there are many parts of the body that are have a mutation. So this is a mosaic individual.
Okay.
It's made up of normal cells and mutant cells because of this mutation that happened during development. Now, here's another example. The mutation could happen after birth. And you would also get a mosaic individuals. So if you have a normal egg and sperm and development, and then during errors occur in a localized area like this, then you also have a mosaic individual. This can happen at any time.
Would you say that this happens with SCN8A individuals as well?
It happens throughout the genome.
Okay,
It's
interesting.
totally random,
Mm-hmm.
completely random. So, can we identify an individual who's a genetic mosaic? That's what you'd like to know. It depends where the mutation arose and which tissues have the mutation. Now, some people are mosaic in their blood, and you see the mutant and the wild type together in the blood. It's very rare. This mosaicism is very rare phenomenon. If it were only in skin precursors, you'd never see it in blood. It would be invisible. Sometimes if you do more than one cell type, you may pick it up here and not there. And if it happened in the germ cells, then it can be passed along, even though the parent is a mosaic. This is all so dependent on chance and so random. So you may or may not know that an individual is a mosaic, depending on which tissues you can actually look at.
Would this kind of help us, and I could be completely wrong here, so tell me if I'm wrong, but we have some parents that have the mutation with a blood test, but they don't have seizures and they're not essentially showing any signs, but then their child has SCN8A and they are affected. So would this kind of be what we're talking about?
That is exactly what we're talking about. They probably had a mutation in a precursor to germ cells. And so the patient, the parent is completely unaffected because this is a brain problem. But they had some germ cells that had the mutation. Exactly.
Interesting. And so some of these parents are being told that their children with the SCN8A mutation, that may not be the cause of their epilepsy because the parents are not showing signs with epilepsy. So that kind of sounds like it could maybe be debated. Is that kind of what, am I understanding that correctly?
Yeah, from what you've said, I would think that's a misunderstanding of mosaicism.
Interesting. Okay, thank you.
Because the parent could have that mutation in blood and in germ cells, but not in brain.
I have a question from the audience. It says it's a parent saying that their saliva was tested. Does that mean that is where it is found? So I think she means like, so her saliva was tested. Does that mean that, okay, yes, I see what you mean. So I'm guessing that she was negative. Teresa, feel free to jump in here. So I'm assuming that your saliva was tested and, it was either negative or positive. So let me give me some more information from that, and then we can jump into that question. Okay, sorry. So her son and she are both positive for SCN8A, and they did a saliva test with her. So I could think
was she positive in blood and saliva?
I don't know. We'll see what she says here in just a second. Teresa, if you're able to, were you tested via blood as well? Saliva only, she said.
And she had it in saliva. Yeah.
Yeah.
But probably it was a mixture.
Was
it only mutant, or was it a mixture of mutant and normal? Well, it would be a mixture anyway for a heterozygous. Yeah, it's one of these situations where it, and the question would be what happened to her germ cells if she could pass it. But it's very likely if she has it in her saliva, then and the child has the same mutation, then it was probably where it came from. So can a mosaic individual pass along the mutation? Only if the mutation is present in the germ cells.
Mhm.
But that you would hardly know, would you?
Yeah, that's what I was, yeah, is there's, to
It
my
would,
knowledge, there's no test, right, for us to be able to test those germ cells.
well, we're coming
Yeah.
to that. Okay, obviously, yeah. And of course, it could just be that one germ cell. The germ cells won't
Right.
necessarily all be affected. So there's really no way.
Yeah.
Here's another way of saying the same thing again. A de novo mutation in an affected child. If the mutation was in that particular egg, in the parent, then every cell in the child will have it. The brown represents cells that are affected by the mutation. So that's a typical de novo mutation. The mutation happened in the precursor, in the egg, in the mother, and the child is affected. But if the somatic mutation occurred in an unaffected parent, somewhere after the germline was, it depends whether it affected the germ cells or not. The parent is patchy, doesn't, for 8A, it's only expressed in neurons. It won't make any difference if it's in these other cells because the gene is not expressed. But if the germ cell had it, then it could be passed along. Does that make sense?
I think so. Jump in there if this doesn't make sense, guys. I feel like I'm asking a lot of questions here, but
Oh, that's great. Yeah. So here are some definitions from the literature. Germline mosaicism, that's what we're talking about. It's a rare phenomenon that occurs when a mutation arises in a germ cell precursor and is therefore present in only some, I can't read the whole screen, but only a subset of an individual's sperm or egg cells. That's what we're all concerned about. Am I a germline mosaic, and could I pass this along to another child? In this case, a variant may or may not be passed on to any given child, depending on whether it's present in that particular germ cell that forms the embryo or not. It's kind of unknowable, isn't it? And germline mosaicism can underline the rare recurrence where a previously mutation that was thought to be de novo in a subsequent offspring occurs when neither parent is known to carry a mutation. So that makes sense, right?
Yeah. So because a lot of us are in that boat, right? That we have this de novo that we feel then, we don't have it ourselves. And so we want to make sure that if we have other children, that they're not going to have SCN8A.
Absolutely.
And so it sounds like it's very rare, but there is still that possibility of reoccurrence, which I think you're going to getting ready to go into right now.
Right. The reason we think the kids are de novo is because the parents are unaffected, and usually it's blood that's tested, and it doesn't detect the mutation. But now you know that the parent could still be mosaic in the germline, and it's fairly rare. I think, there isn't really hard data for how often this happens.
Yeah.
But it may be as much as 3%. So whatever it is, you certainly don't want to take those kind of chances. Well, I don't know. Are you all familiar with in vitro fertilization and pre-implantation genetic testing?
Yeah, I think this is really interesting. So if we kind of back up a little bit, so as we were looking at that last slide and a parent is letting this sink in, because I know the first time we went to our geneticist, they told us it was like this is still rare, but it was like, oh, it's nearly impossible that you'll have another kiddo with 8A. And then about 3 to 5 years later, when we went back in, the geneticist said, whoa, hold on. We're now finding out that there's a possibility that it could be in your germ cells. And so that risk is a little bit higher. Still super rare, but it's a little bit higher. And so now I feel like parents are thinking, okay, and we have different different parents. Some people are like, you know what, it's so low still. We're going to go ahead with natural chances and see. And we have a lot of unaffected siblings in our group, but we have some parents that whether it's anxiety or just that fear of having another kiddo, especially if they're very severely affected, that they want to know, what can we do? Well, is there a way that we can say, absolutely, they're not going to have SCN8A? And that's kind of where you're jumping into this next one with in vitro and pre-implantation.
Exactly. I mean, for people who feel that way, risk adverse, I certainly would, I think. Again, the technology has caught up. And again, the change in what you were told reflects the new information.
Mm-hmm.
We find out it's not as rare as we might have thought. And so this is one possibility, depending on your what you think, but in vitro fertilization and pre-implantation. Now, pre-implantation genetics, comprises a group of genetic assays used to evaluate embryos before transfer to the uterus.
Mm-hmm.
A few cells can be removed. This is really remarkable, from the blastocyst stage. It's a very early stage.
Mm-hmm.
And that was first shown in mice, that you can take a few cells and the embryo develops normally. We're talking about a stage with a couple of hundred cells.
Mm-hmm.
And then if you really want to make sure there wasn't an error in that assay, you can confirm it during your pregnancy with amniocentesis or the traditional prenatal testing and be absolutely sure that a child is unaffected.
Mm-hmm.
So I just have a picture of that here. Pre-implantation genetic testing after in vitro fertilization. So the egg and the sperm are in a culture dish.
Mm-hmm.
And sometimes the sperm is injected into the egg. That's what ICSI is.
Mm-hmm.
And then you get an embryo. You can actually do this as early as the 8-cell stage.
Mm-hmm.
And you take one of those cells under the microscope and you do your PCR test, whatever the mutation you're looking for, exactly what the other child has. So you can do a very precise test. Mm-hmm. And you get several fertilized eggs from this procedure, and you can tell which ones are affected and which are not. It should be 50-50 if depending on the degree of mosaicism.
Yeah.
And then you can transfer an embryo and monitor that pregnancy.
And yeah, I actually had a very close friend that they found out in their first pregnancy that her and her husband were both carriers of cystic fibrosis. And so that meant there was a 25% chance that their pregnancy was going to have cystic fibrosis. And this is, gosh, almost 16 years ago. And so when they decided to have their second child, they did this. And at the time, I live in Missouri, and they had to send after it off to California. It was the only state that would do the genetic testing on the embryos. And they had, after it took several tries, and they have an uninfected child now without cystic fibrosis. And that was before, cystic fibrosis even had a treatment or anything. And so it was very important to them to go this route. Obviously very expensive. I have no ballpark range and of how, what this would cost. Maybe you do, I don't know. But I would imagine that it's fairly expensive. But in your heart of heart, if this is what you feel like you want to do and want to invest in, then that's absolutely a parent's choice. But we wanted to make sure that people are aware of this possibility and that they could bring it up to their OBGYN, or I guess who would they, if they could bring it up to their OBGYN, and then they would probably have to get some type of genetic department involved to figure out the next steps of how to do that. But it's possible.
Yeah, genetic counselors would be the person to discuss it with if you have access to genetic counselors.
Yeah.
They're very familiar with this. This was pioneered in cystic fibrosis,
Oh, well,
which is a very common variant, much more common than SCN8A.
Yeah.
And in fact, it's because cystic fibrosis had such a strong parents foundation that cystic fibrosis was worked on so early, and these options were developed.
That's amazing.
And I think it's used quite a bit by the cystic fibrosis community.
Yeah. Now, Miriam, I did have a question that popped up in the chat. So a parent asked, can IVF cause SCN8A? So my guess is that this particular parent, they went through IVF to get pregnant and their child now has SCN8A. Do we know if that increases the chance, or is it, just as likely with non-IVF ways?
Oh, it would not increase the chance.
Okay. Sounds good. Let's see here. I do have another question. I'm going to read what she wrote on here. My adopted daughter is heterozygous. Her birth mom is a twin. Twin is normal. I believe birth mom is de novo. Birth mom is positive for SCN8A. My daughter's 3 brothers have not been tested. I assume my daughter would have a 50-50 chance of passing it on. My daughter and her birth mom do not have seizures, but do have developmental issues. Trying to follow that without seeing it. I'm sure that's difficult. That's a lot of information.
The mother and the daughter have cognitive problems, but not seizures.
Correct. And so I think, yeah.
So if the mother passed it on, then it would be in every cell of the daughter.
Okay. So,
right. And so that means the daughter would definitely have a 50% chance of passing it on to any child.
Yep, that's kind of what I was thinking too. Awesome.
Yeah.
For following that through. Okay. So
I don't know if this was news, because I think probably most of you were aware of this, but
yeah, and I think that the community might be aware of it. But I think it's something, as especially some of the genetic counselors are kind of changing their stance a little bit on the germ cells as we've learned more, that parents are really going to probably thinking about, do we take the extra step of going through the IVF and pre-implantation genetic testing to assure that. And I guess I should mention too, and stop me if I'm misunderstanding this, but just because you're testing for SCN8A doesn't mean that there might be another rare, something else pop up in one of those 30 errors that we were talking about within the genome. And so, although we can test and say, absolutely, there's no SCN8A, they're not testing every single aspect of the cell to know that something else maybe was in there. Does that make sense? Am I saying that correctly?
That's correct. There are many genetic diseases, and in fact, there are 500 or so in which interventions are possible. And there are tests right now that will test a couple to see whether they have those 500 mutations. They're very inexpensive. These tests are $100 or $200. And then you can know if you both have the same rare variant, then you would want to go down this path.
That's interesting. That's interesting. I know now Stella is 8, so it's been quite a while ago. And I remember that they asked me if I wanted to check for a cystic fibrosis. And I remember it was that, and I can't remember, I think that was it, but it was after I was already pregnant. So you're kind of mentioning like if you wanted to have this test done prior to being pregnant, just to see what you and your partner kind of have going on what's a possibility of your child.
That's a potential these days. But you know, you don't need to do the in vitro fertilization to have a prenatal test. At 3 months, you can do amniocentesis or chorionic villus sampling, and you can test specifically for SCN8A. So you could avoid the expense of the in vitro fertilization. But in that case, you would have to be willing to give up a pregnancy.
Yeah.
That does avoid the expense.
Yeah. Let's see. We have a few more questions. Let's see. If a father passed it on, so in sperm instead of the egg, is it also in every cell of the body of the child?
Yes.
Yes. Okay. I think, were you saying in that one slide that it is more common usually to have it in the egg versus the sperm? Or is
no,
it?
I didn't mean to, no.
Okay, gotcha. So it is, yeah, okay, sounds good. So yeah, because we don't really know, right? Like we can't necessarily trace that back. Okay, so that clears that up, I think.
There are ways, if you wanted to, there are ways, because you can look at these markers, these SNPs, that are nearby the SCN8A gene. And if they differ between the mother and the father, and then the child has one versus the other. But that's very complicated, actually. I take that back. That could be. That's a research project.
There you go. There you go. Let's see. I think the parent was asking who does those tests that you were talking about, as far as if you wanted to get tested prior to pregnancy. That's what we were talking about as far as talking to a genetic counselor, right?
Right. There's a company, they call themselves Counsel, but they misspell it like things do. Counsel, yeah. I guess they spell it COUNSYL.
Okay.
They were one of the first to offer this for $200 or something.
Wow.
I feel that maybe all high school students should test themselves just so they know.
That's really interesting. It could be a really amazing biology project for a biology teacher, right? Like if they could get the or something and then share it. That's interesting.
You know what you're carrying, then you know not to be concerned about.
Yeah, and just to be educated on it, right? So
we've all got defects in the genome.
Yeah. Let's see. I have a couple, and I'm going to kind of try to categorize them in the topics that we're talking about currently. It was asking how much would it cost to test for SCN8A. My daughter's brother has not gotten tested. His adoptive mom wants him tested. Gosh, I think that really just depends on the insurance company that you're working on too, wouldn't you say? And
it's very inexpensive to get a test for a particular gene these days.
Because so it wouldn't,
but
yeah.
If the brother is not affected.
Yeah. I know that's what our doctor, I asked if my son needed to get tested, and she said, honestly, if he's not affected by it, I don't know if you really want to know. And her point too was if he ever wanted to get into the military or something that didn't require a pre-existing, condition, that she's like, if he's not affected, I would just let it lie. But, again, it just kind of depends on the parent and what their thoughts are on that. But,
but you see, the logic is if the sister inherited this, and it would be throughout her body, then if he inherited, it would also be throughout. It couldn't be partial.
Okay.
He couldn't be a mosaic. in which case he would exhibit the symptoms. Doesn't, then he could not have inherited
I see.
mutant gene.
Okay. Yeah, so really no sense in my case, to get that. One parent said that they were told it was about $100. So very, very affordable. Let's see. Another parent said that their daughter inherited from their dad, but she has something on microarray, so modifier gene. He is either mild or non-affected.
We do not know the identity of modifiers that might reduce the severity. So it would not be possible to say this person is mild because they have a modifier. It was just a general way of saying there must be a reason.
Yeah.
A modifier, but we would not know which one.
Okay, let me go through the chat here. So backing up a little bit, a parent was just saying how excited they were about the ASO information that you presented, that they were effective in mouse models. They couldn't, wait to learn more about that, hopefully in humans. And I was curious, with the data that you showed with the ASO, were you treating that ASO with just one mouse model or across multiple?
We've actually tested two models.
Two models?
Yeah, they're both patient mutations. And what it does is it turns down the total expression of 8A. And since we're dealing with an overactivity problem, that just solves the problem by turning down the total. The problem, though, the complication is that you don't want to go too low.
Yes.
But then you get into those loss of function situations.
Madeline talked a lot about that too, and how it was perfectly worded. She said a Goldilocks type mutation where you don't want it too much, you don't want too little. You have this perfect in between that we're trying to go for. So
exactly.
Yeah.
So this is being used in, I don't know if you're all familiar with Dravet syndrome. Dravet syndrome is another sodium channel epilepsy, SCN8A. And they have clinical trials right now because in that case, it's a loss of function problem, and they have an ASO that increases expression. So they don't have to worry about going too low. And those trials are in progress right now.
And 2A has at least one ASO trial currently, and I don't remember off the top of my head if it's gain of function or loss of function. I believe it's gain of function, though.
Yes, I'm not sure, because in that case, either gain of function or loss of function causes different disorders.
Yeah.
Yeah, things are really moving remarkably, not to your, I'm sure it doesn't seem rapid to you.
I know. And that's the thing too, is, as parents, when we see our children seizing and suffering, and it's not fast enough, right? It's not fast enough. But, just in that one slide that you showed us, what has happened since, 1995 and then, 2012. And then it was like, all the things.
Oh, yes, people got busy once it became clear it was a human.
Right.
It's, as I said, it's almost up to 1,000 individuals, and that people all over the globe are aware. So,
well, Stella was, we were estimating about 150 worldwide when she was diagnosed in early 2016. So, I mean, wow. People are getting more genetic testing, and we're able to hopefully treat these kiddos sooner in life and move from there. But wow, I was going to look through real quick and see if we have any more questions. We have a few more minutes if someone wants to pop on and ask something. But gosh, this was a great conversation. And oh yeah, let's definitely acknowledge some people at your amazing.
I just want to acknowledge some of my collaborators, and I have had support from the Cute Foundation. So thank you very much.
Most
and
welcome.
my wonderful little group of hardworking people.
Yes.
Wenji is doing the CRISPR study, and Sophie did the ASO study.
That's awesome. I think I'm trying to remember if we did we hear from your lab last year, maybe for at the gathering? I can't remember.
I think Wenji spoke last year.
That's what I was thinking too. That's what I was thinking too off the top of my head. So awesome. That is great news. Let's see here. How similar is SCN1A to SCN8A, a patient?
Well, you know, they're almost like picture images. They're almost like mirror images, because most of 8A, the mutations cause the channel to be too active. In most cases, many cases, for example, the channel doesn't close properly. So you can understand it gets the signal, it opens, and then it stays open. And so there's too much stimulation of the neurons and they fire. For SCN1A, it's exactly the opposite. Most of the mutations have lost function. So you ask, how could losing function of the sodium channel cause seizures? And the most common explanation is that every type of neuron is unique. And really, the activity of that neuron is a balance of all the channels in it. So you could say, you could invent the word the channelome, you know, it's the channelome, stupid. So the concept for, and there's good evidence for it, for SCN1A, is that that channel is most important in what we call inhibitory neurons. 8A is most important in excitatory neurons. So if you take 8A away from an inhibitory neuron, it doesn't make much difference. But if you take 1A away, then you're reducing the inhibition of the excitatory neurons. And that's the explanation for why the loss of function of 1A is so devastating. The nervous system is extremely complex, as you will know. Right. But they're like mirror images. So for the ASO for 1A, they needed to boost expression. And for 8A, we want to reduce expression.
But not too much.
Not too much, but then you have to consider the relative severity too.
That's true. That would you rather have loss of function type presentation than gain of function?
That's something that everybody will have to think about.
Yeah, definitely. Let's have a couple more questions here. So a parent said, this is a little off topic, but curious if you have any input or information on a splice variant at stop coding.
Whether they exist is the question, whether there are such mutations in 8A.
Let's see here. One more time, but curious if you have any input or information on a splice variant at stop coding. I guess her daughter has the mutation at stop coding.
So, is it a stop coding mutation or a mutation? So every gene at the end of the coding region, it has a stop signal. So does she have a mutation at that stop signal?
It sounds like it.
That's really interesting. Gee.
I have to get back to us.
Yeah, I guess the question is whether there might be some specific way to treat that. It might be one of these situations where a specific ASO, you're familiar with this concept of N of one, N of one. It would require a very special kind of ASO to knock in back that stop codon would be what you would want to do. And of course, gene editing, we didn't even mention that, but that's coming along too. Yeah, that's very interesting.
Another family asked if you're familiar with the research of Jeremy Proctor from University of I'm assuming Michigan, maybe, and what they're doing with SCN8A.
No, I'm not. What are they doing?
Yeah, Tammy, if you want to ask, I've not if I have the name, it is not in my top of my head right now, but so Jeremy Proctor, and I'm guessing University of Michigan, maybe. She said University of M, but I know she's from Michigan, so that might be it. So I'll wait and see if that pops up.
I'll have to look him up. Thank you.
Yeah. It says MSU. I have a hard time with that one. We have an MSU in Springfield,
Michigan State, Michigan State University. Oh, that's good to know. I will definitely look him up.
Yeah, me too. Absolutely. Let's see here. What else we got here? Other questions? It sounds like a parent is saying that, how do we increase testing if there are developmental delay issues? When young neurodevelopmental doctors didn't think testing would change the approach.
Well, there are many physicians who are very practical, and they say if the diagnosis is not going to make a difference, then why do it? But with treatments developing so rapidly, I don't think that's a very strong argument. And if you can go ahead and get the testing anyway, like right now, they're not testing adults, and that's kind of a mistake, I think, in limiting what we understand, what we know about the disorder.
Yeah, seeing some of those longitudinal type presentations, and yeah, I can see that. Well, this has been a very lovely discussion. I was going to see if anybody had any more questions before we wrap up. But wow, thank you so much, Miriam, for coming. I really, really appreciate it. Oh, we got another question. How do we get our doctors in touch with the research projects for our children to be involved?
For clinical trials?
I think so,
for research. Well, there's an international SCN8A
absolutely
society now.
Yeah.
And there's Michael Hammer's SCN8A.net. And the answer is through these parents groups that are doing longitudinal studies and collecting all the data you need before you can do a clinical trial. It's very important, and every family should get involved if they can.
Yeah, absolutely. And I think, a lot of these things that we just chatted about too are still, in mouse model research, and not in humans. And so, I think even though we've came so far, there's still a big jump that we need to take before we can get seeing this in humans and then getting these things in clinical trial. And so that's what I think.
The importance of getting into these registries now is that when the clinical trials are started, they can find you.
Yeah, exactly.
If you don't sign up, nobody knows you'd like to be involved.
Yeah. So I think, both groups are amazing at trying to get the word out of SCN, about SCN8A, and that we exist and that we have this strong, amazing community between both groups, both organizations, and we're both committed to moving this forward. So I think if we keep sticking together and keep moving forward and being the squeak wheel, then we're going to get that. It is really remarkable that every single conference I've been at the last 2 years, SCN8A is brought up. I see it on the screen, and that's amazing to know that we're out there. Let's see. Looks like he is studying. Oh, I think this or Jeremy Proctor. He is studying the mutation who we don't know how they are working, or how they work. So yeah, we'll definitely look into that. And it looks like Miriam has wrote down that name too.
So the parents,
and I think we
made a huge difference. They made the difference in cystic fibrosis, and they're making the difference in these sodium channel mutations.
I'm really excited too. We have a couple of new faces and names that are going to present at our 9th annual gathering coming up too. And just to make sure, yeah, everybody knows that we're out there and who's working on what too. So that way, you know, any type of collaboration or connections we can make too is beneficial. And a parent just said, "Thank you, Miriam. You are great," which I will have to mirror that. I just appreciate not only you know, what you've done here today, but what you've done throughout your entire life. I mean, you've essentially, you know, dedicated your life's work to SCN8A and other sodium channels, and we just appreciate you so very much and what you've done for our kiddos with the work that you've done.
Thank you so much.
You are
Thank you,
most
everybody.
welcome. All right, guys. Well, thank you for all of the parents too that have shown up during this series. We've had a remarkable attendance this year, and it really just goes to show, as parents, we really want to know and understand the science behind SCN8A and you know, how much passion we have for us. And I like not all of you, whether it's virtually or in person, for our 9th annual clinician, researcher, and family gathering. And I can't believe I'm saying this, but it's like in 2 weeks. So I'm going to jump right off here and work on my list for the gathering. And it's going to be a great time, whether you're there virtually or in person. But can't wait to give everybody hugs and see everyone. And one more shout out to Platinum sponsor Neurocrine Biosciences for their sponsorship. We cannot do these educational series without them. So thank you. All right, everybody. Well, thanks for attending. Have a lovely day, and we will see you in Orlando. And thanks to you, Miriam. Thank you so much.
Ciao.